Our first speaker today is Dr. Michael Aschner. Dr. Aschner is a professor of pediatrics and pharmacology and the senior investigator at the Kennedy Center of Research in Human Development. He serves as the Harold and Muriel Block Chair for Molecular Pharmacology at Albert Einstein College of Medicine. He has served on numerous toxicology panels, including the Institute of Medicine, the U.S. Environmental Protection Agency, and the Center for Disease Control. He is also a member of the Neurotoxicology and Alcohol Study section. His research focuses on regulators of mercury uptake, mercury-induced oxidative stress, mercury neurotoxicity, and metal-induced neurodegeneration. Please join me in welcoming Dr. Michael Aschner.
I'm going to talk to you about mercury, and I'm going to talk about exchangeably about all three different species because they have common neurotoxic effects. The first slide is just to show you some of the effects of mercury, and I'll have a couple other movies that will establish the neurotoxic effects. I was fortunate actually to visit Minamata, which some of you may be familiar with. I was there a couple times. This is the museum in Minamata, and what happened in Minamata was that mercury that was used by a factory there, Chizo, at the time, and the factory is still there, not using mercury anymore, but it's still there nevertheless. This is a small village, fishing village in the southern island of Japan. And what they did is basically dump mercury into Minamata Bay in Japan. And this mercury accumulated in sediments and then got into the fish and eventually was consumed by human beings. And these are some of the devastating effects that you find with mercury. It has developmental effects. It has effects in adults. One of the interesting things actually that happened there was that the fishermen found that when they actually moored their boats at the site where the effluent from the factory was getting into the bay, they didn't have to worry about cleaning their boats because everything that was growing underneath the boat was basically destroyed by the mercury. So they used to park their boats or dock their boats downstream to the effluent that was released from the factory. Now, I have a couple movies. I hope you can bear with me, and I hope that it works. The interesting thing in Minamata was that the first thing that actually alerted individuals to the fact that people who were consuming the fish from the bay were becoming sick was when they looked at the cats. And this is just one example, and I hope you can watch it. But this basically shows the effects of mercury at high levels on cats. You can see they're ataxic. They cannot bear their weight on their hind limbs. They're disoriented. If you put them actually on a beam, they will fall down. So the effects are devastating, and this was the first thing that alerted the individuals to the toxicity of mercury at the time. There was another epidemic. This was in the 60s, late 50s and early 1960s. The next one was in Iraq, where they used grain that was actually shipped from Mexico that had mercury in it to the Iraqi farmers, and everything was labeled in Spanish. Obviously, people in Iraq who speak predominantly Arabic didn't know what was labeled on the grain, and the grain arrived in Iraq post the seeding season. So rather than seeding it and growing their wheat and flour, they decided to directly use the wheat to bake their pita bread. And this is, again, a movie that is not fun to watch, and I apologize to some of you, but these are the labels. mercurial poison. It was labeled. They made the pita bread. And the next slide, or as we progress with the movie, you'll see the devastating effect that the mercury has on a specific child. This is a child that's exposed to mercury. Again, you see ataxia, you see inability to touch the nose in this example. This individual has problems with both sensory and motor function. Here he is trying to put some food in his mouth. You can see the cerebral palsy type of effect in his hands. They're flexed. And I think I'll stop it here because the rest doesn't really add very much. So I'm here to talk to you about mercury. And what I hope I can do over the next 45 minutes to an hour is to give you some appreciation for the different chemical and physical properties of mercury. And again, I'll talk about the 3 different species, and I'll emphasize specifically why in your case mercury vapor is more important and how it behaves relative to the other species. We'll talk about the environmental transformations that determine the bioavailability of mercury. And then we'll talk about some common mechanisms of mercury toxicity. We'll talk about different outcomes with respect to mercury in its different forms of exposure. And I'll spend most of my talk actually talking about genetic predisposition to mercury. And some of the examples relate to methylmercury because I work more with methylmercury, but nevertheless, there's a lot of commonalities between methylmercury and the inorganic mercury. So I think everything that I'm going to tell you about the methylmercury applies to the mercury vapor as well. So these are the different forms of mercury. I don't need to say anything about amalgam here. Thimerosal, I probably don't have to say very much about it either. It's been in the news. It's not being used anymore in the United States in the vaccines, but unfortunately, when vaccines are shipped to developing countries in big vials, it's a lot cheaper than doing the individual vials. And in order to prevent bacterial infection in those vials, they still use thimerosal. So, and I think there's some eye lotions and other preps where thimerosal is included. And methylmercury, as I mentioned in my talk already, from the example in Minamata, there are other examples, there are other cohorts that are actually being studied now in the Seychelles Islands, in the Indian Ocean. There's another big cohort that's being studied in the Faroe Islands. And the effects of methylmercury are being studied there. The consumption of methylmercury is mainly through dietary intake of fish. Now, one important thing that we actually don't consider very often is what happens when somebody has high fish intake and they're also exposed to mercury vapor, for example. That's really not being looked at all. So this is the geochemical cycle of mercury. Most of the mercury is, or a lot of it is man-made. It's anthropogenic. It's coming out from power plants. Mercury then is transported in the environment. The mercury vapor gets oxidized. It forms predominantly inorganic mercury, which then sediments in waterways. And it's interesting the way that it's getting methylated. There are many bacteria in the water that, in order to defend themselves, they put a methyl group on the inorganic mercury to form the methyl mercury. By putting the methyl group on the mercury and converting into methyl mercury, they are able to excrete the mercury. So for the bacteria, it's a defensive mechanism. They can excrete the mercury, but then what happens is that the methyl mercury starts accumulating in fish, and as you move up in the hierarchy of the fish in marine, water, what you find is that the predator fish, which are the fish that we like to eat, swordfish, tuna, and so forth, end up with the highest levels of mercury. And it's not uncommon, actually, to find mercury in fish at levels which is 10, 100-fold higher than the mercury level in the water. How much mercury is going to be in the fish depends on where the fish is caught. Again, that's dependent on industrial properties, where the fish are being caught. In the northern hemisphere, for example, where the industrialized world is much more developed than in the southern hemisphere, levels of atmospheric mercury are about 2 to 3-fold higher than in the southern hemisphere. And there's hot spots, which is probably related to how much mercury is in the sediments themselves. I don't know how well you can see it, but this basically just shows the target organs, the signs of toxicity. It's hard to read from here. The systemic clinical signs, and these are the different species. So this is mercury vapor. This is the inorganic divalent mercury, the methyl mercury, and the ethyl mercury. And I think without going through the whole slide, what you can see is that there are differences in the toxicity of the species. And I'll talk about it in the next few slides for the reason for it. The mercury vapor affects kidney. It also affects the nervous system because it's a vapor, so it can readily cross different membranes, and it can readily accumulate in the brain. You don't see that with the divalent mercury, mercury in the plus oxidation state, because there are no good transporters for this species, so it does not get into the brain. Most of the inorganic mercury goes into the liver, and then it accumulates in the kidney, and most of the effects are on kidney function. Methyl mercury, on the other hand, is very well absorbed. I'll talk about it. There's specific transporters for this species of mercury, so it very readily gets into the brain. And the main concern with the methyl mercury is on the nervous system, and the same holds pretty much for ethyl mercury, which is the mercury species that's being used in thimerosal. So a little bit about why these species are different in terms of their toxicity. So this species is probably the one that's most important for you. This is the mercury vapor. We obviously inhale it. It's originating from amalgams, unless you work in a mine or somewhere else. It's a vapor, so it's very diffusible. It gets very readily through membranes, and this black line here is basically the membrane, the alveolar membrane, separating the air from the blood. So mercury vapor is the diffusible gas. It gets into the blood. In the blood, it gets into the red blood cells. And interestingly, a small portion, and this also takes quite a bit of time, mercury vapor is converted to Hg++, which, as I said before, is the species that doesn't get very readily into the brain because there's no transporter for it in the blood. So most of it goes into the kidney and it's excreted. What's interesting about these species, and this is anecdotal, I'm not sure you want to use it in the clinic, but there have been studies actually that show that people who drink a lot of alcohol, their body burden of mercury vapor goes down. The reason for that is that alcohol, like mercury vapor, is a substrate. Mercury vapor is a substrate, like alcohol, for catalase and hydrogen peroxide. And when you have hydrogen, when you have a lot of alcohol on board, what you're doing is you're reducing the conversion of mercury vapor to the Ag++. So the amount of mercury vapor that stays in the blood is increased. And since it's inhaled and exhaled, if you work in a mine and you drink alcohol, your body burden of mercury ends up being lower. And there have been studies to show that. So mercury vapor is a substrate for catalase and hydrogen peroxide. It gets oxidized. The same happens in the brain because it takes time, as I said, for mercury vapor to undergo this process. Some of it readily crosses the blood-brain barrier, which is a very tight membrane that separates the brain from the blood. And once it's in the brain, it gets converted to the inorganic species, and then it starts wreaking havoc. And that's the reason why mercury vapor is toxic to the nervous system. Hg++ itself actually does not cross this barrier. So if somebody is exposed to inorganic mercury in the divalent form, that's the reason that the brain is not a target organ. And this is also shown on the next slide. So this is the metabolism of the inorganic mercury. We get it in the food. The absorption of inorganic mercury is very low. It's only about 5%, so it doesn't get into the blood. And therefore, most of it goes into the liver. Subsequently, it goes into the kidneys, and it's excreted in the urine. So for inorganic mercury, actually, urine is a very good surrogate or biomarker and can be used to assess exposures to mercury. It's not a very good measure for mercury vapor because most of it gets into other tissues, and then once it's converted to the Ag++, it's being trapped. It's interesting because it's such a toxic substance, actually. In the old days, some of you may be familiar with, there was a drug that was called Mercury. Mercury was used as a diuretic because mercury, once it gets into the kidney, it blocks ATPase, so it blocks the active transport of sodium across this membrane, and you get diuresis. And now, obviously, it's been removed from the market. And the last one, no more burdening you with too much metabolism, is the methylmercury. The reason that I'm doing it is just to show you that there are chemical and physical differences. The mercury comes primarily from fish. It gets into the intestinal tract, and actually we have an enterohepatic circulation for mercury, for methylmercury. The absorption rate of methylmercury is very efficient. About 95% of it gets into the bloodstream, unlike inorganic mercury, which I said is about 5%. So it gets into the liver, and then it goes through the portal circulation, it goes through the bile duct back into the intestinal tract. That's the enterohepatic circulation. It's interesting because people who, for example, receive certain antibiotics where you wipe out the gut bacteria end up, again, having higher concentrations of mercury body burden. The reason for that is that in order to break this cycle, we have bacteria in the gut which do the opposite of what I told you is takes place in waterways. These bacteria are actually able to demethylate the mercury. So as the methylmercury circulates here in the gut, it gets demethylated. You end up with divalent mercury, Hg++, which has a very low absorption rate, and that mercury is then excreted in the feces. So one consideration about mercury and body burdens of mercury is whether an individual actually takes antibiotics. Antibiotics can interrupt this cycle, and the enterohepatic circulation continues unabated. Once mercury is in the blood, it actually conjugates with one of the essential amino acids, which is methionine, which I'll show you in the next slide. And methylmercury methionine can then get transported into the brain. And that's the reason why the brain is the primary target for methylmercury. We call the transport of methylmercury across the blood-brain barrier molecular mimicry. What you see here is the methylmercury. This is a cysteine molecule here, which is found in the blood. When the methylmercury binds to cysteine, it forms a methylmercury cysteine complex, which is shown here. And the methylmercury cysteine complex is very similar to this neutral amino acid, methionine. You can see there are very small differences. Basically, you just have the mercury trapped here, and you don't see it here. It turns out that this molecule here, methylmercury cysteine, and mercury has very high affinity for cysteines, binds to the neutral amino acid transporter, which is known as large amino acid transporter, LAT1. And basically, it fools this transporter to make it believe that this is another molecule of methionine. There have been studies, we've done some studies and others. If you use very high concentrations or doses of methionine, you can actually block the transport of mercury into the brain. So this is a competitive transport between this complex methylmercury cysteine and an endogenous molecule, an essential molecule, methionine. I'm going to shift gears a little bit, and I'm going to get now into the genetics. And you work with humans. A lot of the work that I do is actually with very small worms. But hopefully I can convince you that what we learn from these worms is very applicable to humans. So these are worms. They have a very short life cycle. We use them because the power of genetics. And I think that's going to become important throughout my talk because, as I said before, everybody in this room is different. And because we can use genetics in these worms, we can modify different genes, and we can ask specific questions about whether these genes actually bestow sensitivity to mercury in specific strains of worms. We can freeze the worms, which makes it very easy for us. We can go away to Denver, go back to New York, and work with them again. And the nice thing about them is that, unlike us, we have billions and billions, trillions of cells. They have only 959 cells. And for those of us that are interested in the nervous system, it's even more exciting because they only have 302 neurons. And we can tag these neurons with specific markers. Commonly, we do it with a green fluorescent protein. So we can actually visualize the worms under a microscope in their living state. We don't have to anesthetize them or anything. We can look at them and we can count different cells, and we can ask questions about what's the role of a certain gene in bestowing toxicity, how can we protect the neurons from mercury or other compounds, and so forth. So surprisingly, we have about probably 25,000 genes. In this 1 millimeter creature, there's 19,000 genes. So they're not that different from us. We can actually do even behavioral studies in these worms, which I don't think I'll show you today. But there's about 70% homology between the genes in the worms. And that's important because if you're going to query the function of a gene, you want to make sure that that gene has similar structure to the mammalian gene. And we can also ask questions about genes actually that they don't express. We know, for example, that people that develop Parkinson's disease have mutated alpha-synuclein. Alpha-synuclein is not expressed in the worms. So what we can do is take human alpha-synuclein or human mutated alpha-synuclein, we can express it in the worms and then ask specific questions about how does the alpha-synuclein affect the worm's behavior, how does it affect the wealth of the different neurons, and so forth. Now, what I'm going to try to relate the mercury to is Parkinson's disease, and that's true for mercury vapor, methylmercury, any type of mercury. As you know, there are different types of Parkinson's disease. There's one that's the familial type, the Michael J. Fox type of Parkinson's disease. I believe he was about 28 years old when he developed Parkinson's disease. That's inherently genetic, so it's 95% genetic, if not more than that. The other sporadic, or what you might have heard before, idiopathic Parkinson's disease is of unknown etiology, idiopathic. And that's only about 20% genetic, which means that 80% of the contribution derives from a different source. And in this case, it's the environment. And there have been many compounds that have been associated with Parkinson's disease. One of them is actually mercury. Now, I'm showing you this map of the United States because if this was a genetic disease, you would have expected that the color, which is basically the prevalence of the disease for a given amount of a population, would be the same. You'd expect it to be either red or green or yellow throughout the United States. But as you can see, it's not. There's hotspots. Interestingly, the Northeast, not so much in California, and in the Midwest, some areas, the frequency is higher than in other areas. So the point is that idiopathic disease is not a genetic disease. It's only about 20%. 80% of it is derived from the environment. And what I want to focus next is on the role of mercury in this disease. So a causal relationship between mercury and Parkinson's disease has been noted. Both Parkinson's disease, and I apologize for some of these typos, and mercury poisoning present with resting tremors and alterations in motor functions. I didn't talk very much about the symptoms of mercury, but I think you're familiar with this. Several studies also have shown association between fish consumption, so this applies more to the methylmercury, but also occupational exposures to mercury and increased prevalence of Parkinson's disease. And the last bullet here is that low serum mercury levels are associated with a decreased risk for Parkinson's disease, and serum mercury levels are significantly higher in post versus pre-Parkinson disease diagnosis. And actually, most of these studies have been with inorganic mercury, with a vapor, not with the methylated form. Now, one of the genes that is known to be associated with Parkinson's disease is referred to in mammalians as Parkin, or PARK2, and it accounts for about 50% of the cases of Parkinson's disease, the familial type. I'll show you in the next slide what this gene does. It's basically an E3 ubiquitin ligase, which is, as I said, I'll show you in the next slide. And it's the importance of this gene is that it regulates the function of the mitochondria, which are very important in supplying energetics for the cells. So if you have alterations in this gene, you might presume there might be some consequences to the mitochondrial function, and if you have loss of mitochondrial function, everything goes awry. You induce more oxidative stress, and the cells are dying. So what we decided to do was to look at what are the effects of mercury in worms on animals that have mutations or where we have knocked out this gene. And as I said, it's known as PARK2 or Parkin in mammalian systems. In worms, I'll refer to it as PDR1. And basically the hypothesis here was that exposure to mercury and alterations or knockout of PDR1 will exacerbate the toxicity of mercury toxicity and damage the dopaminergic neurons. So the dopaminergic neurons are important because those are the neurons that are damaged in Parkinson's disease. When you lose 80% of your dopaminergic neurons, you start seeing the typical motor and behavioral effects that are associated with the Parkinson's disease. And again, why is this important? And I think hopefully I can emphasize it later, is because each of us here in this room express different levels of PDR1. So the question becomes, if you're a good expressor of PDR1, are you less sensitive to mercury? And if you are a poor expressor of PDR1, is mercury going to exacerbate the toxicity? And in this case, the endpoint that we're looking at is functionality as it relates to the dopaminergic system. So this is just one slide to show you the functions of PDR1. As I said, it's an E3 ligase. What it does basically is take proteins, and as the proteins get to this gene or gene product, it's being ubiquitinated, which is shown here. There's many ubiquitin groups that are put on the protein, on the target protein, and then this protein moves on to the proteasome, where it's ultimately being degraded. And this is the way we actually get rid of most of our proteins, by ubiquitination. So the question becomes, if this gene doesn't function well, are we for some reason altering protein levels? Are there changes in proteins that might be important in transporting mercury, for example? Might it lead to increased burden of mercury in the body, in these worms, and so forth? And these are the kinds of questions that we ask. So what happens, first of all, in terms of lethality? Just for orientation, N2 are normal worms, normal people. PDR1 knockouts are the worms that don't express the PDR. So you would presume these worms would be much more sensitive to environmental agents, and if you could actually look at tremor in the worms, these worms would be showing you tremor. So the first essay that we did was just basically look at lethality. And again, for orientation, this is the number of live worms here, and what we're looking at here is the dose of mercury. And the black line is normal worms, N2s, and the knockouts are here. And the way we look at different parameters in toxicology, at least crude ones, is LD50, lethal dose 50. What does it take to kill 50% of the worms? What dose does it take? So it would be about here. This is 50%. And you can see actually that for the whole curve, there's a left shift in the PDR knockouts. What does it mean? It means that for the same dose, you actually kill a lot more worms that don't express the PDR versus those that are normal and express the PDR. So the LD50 is lower in the PDR knockouts. That is, with lower dose of mercury, you kill more worms. Okay, so they're more sensitive. If you look at the lifespan, and I didn't mention it before, these worms live for about 3 to 4 weeks. That's their normal lifespan, which is nice for those of us that do research, because unlike studies in rats or other species, we could do it fairly quickly. These are the normal worms. If we treat them with mercury, we actually don't see, these are 2 different concentrations of mercury, relatively low concentrations. There's no difference. These are the PDR worms, and these are the normals, and you see that with 10 and 20 micromolar, again, there is a shift to the left. So the worms that don't express the PDR1 are much more sensitive when you hit them with relatively low doses with mercury. Their lifespan goes down, again, showing increased sensitivity to mercury. Interestingly, when we look at the mercury levels, and here we use ICP-MS in the wild type, in the normal worms, and in the PDR1 knockout worms, what we find is that in the absence of mercury, the mercury levels are about the same. But when we treat with mercury, the PDR knockouts actually have much higher levels of mercury. So this suggests that there is something going on in terms of ubiquitination and the degradation of different proteins in the PDR knockouts, which, as I said to you before, is one of the functions of this protein. So next, we decided to look at the levels of these transporters. So the terminology is a little bit different in the worms. It's amino acid transporter 1, 2, and 3. And the important thing to note, and without too many details, is that the levels of these transporters, AAT1 and AAT2 specifically, in these 2 slides here, are actually much higher in the worms. So this suggests that when PDR1 is knocked out, or when you have mutations in this protein, the ability of the cell to degrade the mercury transporter goes down. And consistent with the increased expression of the transporter, you find higher levels of mercury in these worms, as I've shown you in the previous slide. So PDR knockout, the lethality goes up, lifespan goes down, the amount of mercury goes up, and this is associated with increased expression of the transporters. So C. elegans is an ideal model to study the interaction between loss of function of a gene and mercury toxicity. And Mercury, as I've shown you, is transported by the amino acid transporter, and genetics or altered genetics can affect the toxicity, and I've emphasized this specifically with parkin or PDR1. Now, another gene that's very important in terms of regulating outcome of toxic exposure is known in mammalian systems as NRF2. It's called SKIN1 in the worms. And this is just a scheme to show you basically what happens. When there's a lot of electrophiles, when there's a lot of reactive oxygen species generated, such as by Mercury, what you get is that KEAP1, which is a protein that's associated with NRF2, gets oxidized. This protein has a lot of SH groups for which we know Mercury has very high affinity. The KEAP1 gets oxidized. It dissociates from the NRF2 or SKIN1, and the NRF2 moves into the nucleus. It binds there to a consensus sequence, and then you get upregulation of different proteins that are associated with antioxidant effects. So this is a sort of a positive effect. You want this to go on. When we treat, in this case, astrocytes or microglia, from rats, and these are Western blots, you can't see them very well, but basically you see that in the presence of Mercury, we get a very quick response in the microglia. So it seems that the microglia are the first line of defense when animals are exposed to Mercury. You get upregulation of the NRF2 or the SKIN1 in worms. The reason for that is that you want to produce a lot more antioxidant effects, okay? Or antioxidant proteins. And this is shown here. The red labels nuclei, the green is the NRF2, and when you hit the cells with Mercury, you can see that there's a lot more yellow color, which basically is the combination of the green and the red, signifying that the green, which is the NRF2, moves into the nucleus. That's why you get the yellow color. You see the same in microglia, and you see it here in astrocytes. If you look at worms, where you have knocked down PDR1, okay, so we're coming back to this important gene, you can see that for the same amount of mercury, these worms produce much less SKIN1, okay? Which means that the ability of these animals, which have alterations in this protein in PDR1 or parkin, is reduced. So you would presume that for the same amount of exposure to mercury, the animals that have altered parkin or PDR expression are much more sensitive. So the combination of exposure to mercury and PDR1 has significant effects on oxidative stress. In response to mercury exposure, PDR knockouts show altered activation of the SKIN1 pathways, which I've shown you in the previous slide. So basically, for the same amount of oxidative stress, and they do have the same amount of oxidative stress, they will have a lessened ability to mitigate oxidative stress, allowing basically for this brush fire kind of effect to continue to go on unabated. In other words, for the same level of ROS generation of PDR1 is likely to be far more sensitive to the oxidative stress, as they will be unable to mount a proper antioxidant effect. So again, if you translate this to mammalians or to humans, people who have lessened PDR gene expression are much more likely to be sensitive to mercury exposure because they are unable to mount the same amount of SKIN1 response. Less of it is going to move into the nucleus, and for the same amount of oxidative stress, or higher in the presence of mercury, their ability to generate those antioxidants in the form of glutathione or methylthionine and so forth, hemoxygenase, is going to be reduced. The next thing is trying to relate this specifically to dopamine, because that's where Parkinson's disease occurs. And we can actually study it in worms. And again, without going into too many details, this is a schematic of a presynaptic neuron that makes the dopamine. This is the postsynaptic neuron. And this is the terminology. I'm not going to go into it. I'm just going to say that, amazingly, the worms have exactly the same machinery that we have. They make the dopamine, they synthesize it, they release it. The postsynaptic receptors are exactly the same. And if you recall, I said in the beginning of my talk, we can actually take green fluorescent proteins and we can label specific proteins that are associated with this specific machinery and then visualize under the microscope specifically dopamine neurons. And the worms have 8 dopamine neurons. 2 of them are in the back or in the tail, and 6 of them are in the head. These neurons are important in terms of feeding behaviors, in terms of motion, and so forth. And what I'm going to show you in the next slide is how we can visualize these neurons. So we take this green fluorescent protein, we put it on a promoter for dopamine transport, which is specifically expressed in dopaminergic neurons. And we look at the worms. So they're tiny. They're about 1 millimeter. This is the way they look. This is the head, and you see 6 dots, you have to believe me, in the head, and 2 of them are in the tail. And if we turn the lights off here, there's 6 neurons here, and there's a couple neurons here. So we can now treat them with mercury vapor, we can treat them with methylmercury, and we can ask specific questions about what happens to these neurons. And furthermore, we can relate the effects of these different compounds to the genetics of the worms, because we can do it within the context of animals that have altered skin 1, so they're unable to mount antioxidant responses. We could do it within the context of PDR knockouts, so we can ask all kinds of questions about the interaction between mercury and the genetic milieu. So in this case, what we looked at specifically is how are the mitochondria related to this. And I know, again, this is complicated, and I'm not going to say very much, but one of the important compounds that is endowed with regulating and maintaining mitochondrial function is NAD. And the NAD redox shuttle is shown here. There's a couple of them. One of them is here. One of them is associated with glycolysis. One of them is associated specifically with the electron transport chain. It doesn't really remember. Just know that NAD, NADH is important in terms of regulating mitochondrial function. So what happens to the neurons in the presence of mercury? Instead of using green fluorescent protein, now we use a red cherry protein. It doesn't matter. So these are the 6 neurons, the dopamine neurons in the head of the worm. If we expose them to mercury, you can see neurodegeneration of these neurons. You see the amount of redness goes significantly down. If we pre-treat the animals with NAD, we can actually rescue them. Oh, this is NAD alone. This is NAD with methylmercury. The NAD can completely rescue the effect of methylmercury. So these worms look identical to control. Mercury itself reduces the levels of NAD. So this is controls, and this is 2 different concentrations of mercury. It goes down. If we pre-treat the animals with NAD, they look exactly like controls. This is actually a behavioral study. So what happens to these worms? The worms are like us. We feed them bacteria. They like to eat E. coli. You put them in a plate. They move. You expose them to food. They slow down, akin to us going to, I don't know, McDonald's. We get the food, we sit down, we stop walking. So there's a difference between the amount of body bends in these worms in the presence of food and in the absence of food. And we can look at this difference. And since the feeding behavior is driven by the dopamine, we want to see that there's a big difference in the absence of food and in the presence of food. And it usually goes down by about 10 for a 20 to 30 second scenario where we look at the number of body bends. So These are control worms without mercury. You see when we expose them to mercury here and here, the number of body bends goes down. So the delta goes down, which essentially means that they don't slow down. The number of body bends stays pretty much the same. If we give them the NAD, we can actually recover this sedentary period. They move less. And this is very consistent with what I've shown you in the previous slide, or a couple slides back, where dopamine levels are reduced, since this behavior is driven by dopamine. So by giving them NAD, we can rescue dopamine fluorescence, which suggests that the number of dopamine neurons is intact, and they slow down when you expose them to food. So there's a functional, actually, readout of this effect on the dopamine neurons as well. I mentioned the mitochondria before, so we can also look at mitochondria by, there's a specific stain that labels the mitochondria. When it becomes more red, it means that the mitochondria are depolarized and less functional, and so these are the controls. This is in the presence of mercury, 2 different concentrations, and when we treat with NAD, pre-treat, you could see they look like control. So again, restoring mitochondrial function in the presence of NAD can rescue from loss of function of the mitochondria. And I've seen some actually booth in the exhibit on glutathione, and it's very consistent with what I'm showing you here. If you treat with mercury, levels of glutathione go down because it's the primary antioxidant that we have. As you generate more oxidative stress, you're consuming the glutathione. So treatment with methylmercury leads to, or mercury itself leads to reduced glutathione levels. And when you treat with a NAD, you can restore glutathione levels in the worms. So, to this point, we established aberrant mitochondrial function in mercury-induced neurodegeneration, linking it to potential causality in Parkinson's disease. I've shown you that NAD can alter mitochondrial function in readouts of multiple endpoints, and by treating with NAD, we can recover mitochondrial function, and we can restore the dopaminergic function. Another protein that I want to mention is metallothionein, which is also known to alter mercury function. Metallothionein is very rich in cysteines. About 33% of all the amino acids in metallothionein are cysteine. So it's like a sponge for mercury, just like glutathione. So when you treat or when you're exposed to mercury, it binds to the mercury, and it's then excreted. Genetic susceptibility to high body burden has been suggested in autism, as you, I'm sure, know. And one of the genes actually that is known to be associated with autism is metallothionein. So the idea here that SNPs in metallothionein or single nucleotide polymorphisms in this gene, metallothionein, might increase the predisposition to autism. You can study autism in worms, but this just shows you, in terms of genes, that there is a lot of overlap. This is known as a Venn diagram between different genes upon exposure to mercury in the form of methylmercury and inorganic mercury. And this is taken from one of Jonathan Friedman's manuscripts. These are concentration of mercury in live mice following exposure to mercury, and you can see that the levels go significantly up. And what I'm showing you here is, going back to the worms, is that metallothionein null animals, so animals that don't express metallothionein, which is down here, are significantly more sensitive to mercury. So this is, again, percent of worms that are alive upon exposure to mercury. The blue is the control. And I think that you can see that animals that are empty null, where we have knocked out the different genes, MT1, MT2, or both of them, are much more sensitive to mercury. Again, translating into humans, this would suggest that if you have certain SNPs or polymorphisms in metallothionein, you might be more sensitive to mercury because the buffering ability against the inhaled mercury or the mercury that's in the blood or is already in the tissue goes down, given the lack of this sponge that can bind to mercury. Now, I want to wrap it up. I have 5 minutes, so I want to wrap it up all together and make it sort of more germane again to how human genetics might affect all of these effects of mercury. So more recently, we worked with what's known as the BXD recombinant in mice cohort. This is a cohort of 120 different mice strains that have been inbred. They've been phenotyped and they've been genotyped. So there's tons of data about these mice, and we know about the expression of every protein in these animals. And one of them is SKIN1 on NRF2. You may recall NRF2 is the protein that drives the antioxidant response. So what I'm showing you here is that the levels of NRF2 in these mice are very different. And it's true for the people sitting in the room as well. Some animals, such as the BXD21, are what we call good expressors of NRF2, and BXD84 are bad expressors of NRF2. So you might postulate that these animals here might not be endowed with a very efficient system to fight increased oxidative stress, no matter what the source is. If you look actually at these animals, even in the absence of mercury, which is fascinating, this is a paper that we published a couple of years ago, you find that the BXD84s, which are the animals that have bad NRF2 expression, show different behavioral effects. So this is even in the absence of mercury. They exhibit social avoidance behavior, which is associated with emotional disorder, even in the absence of mercury. This strain also spends less time in elevated open spaces during the elevated plus maze test, which is associated with anxiety-like behavioral alteration. So you can imagine, actually, if you take these animals and you throw in mercury or some other compound, you would expect an additive effect, if not a synergistic effect. They would likely be much more sensitive than control. And indeed, this is the case. This is a study that was taken from a colleague of mine, Byron Jones. These are different strains that he looked at, but interestingly, one of them is the BXD84, which is the one that has less ability to combat oxidative stress. And what you find in this strain is that when you expose them to MPTP, which is basically it was used, I don't know if some of you may remember, in the Bay Area, San Francisco, in the 80s. It was a designer drug. Basically, it's a pyrimidine compound. It was used for, young people used it to hallucinate, and it turned out that many of these people actually developed Parkinson's disease in their 20s or 30s, and that's very uncommon unless you have the familial type of the disease. So when you take the BXD84s and you expose them to MPTP, they're much more sensitive, again, likely because they are unable to mount an adequate or optimal response to, this is not mercury, this is MPTP, but you might think that in the presence of mercury you would see the same effect. So my final comments are that common mechanisms of mercury toxicity include targeting of neurotransmitters. I've shown you dopamine effects. We know well from different types of studies in many, many different species that mercury will increase reactive oxygen species. There's altered antioxidant response, as I've shown you. And we know that one of the organelles that's probably most important in executing this toxicity is the mitochondria. If you add NAD, you can rescue the animals. You could do the same with high expression of metallothionein. High expressors, which I didn't show you, of SKIN1 or NRF2, which is the antioxidant response element, also are protected against mercury. So altogether, genetic predisposition And I want to emphasize we are all different from each other. And this is why studies in BXD, recombinant inbred mice, or in genetic mutants are important. Genetic predisposition and SNPs in different genes may govern, or will govern actually, the toxicity of mercury and its neurodegenerative effect. And hopefully I convinced you that glutathione, PDR1, parkin, metallothionein, just to name a few genes, are going to be important in determining the outcome of exposure to mercury. And with that, thank you for being here early at 8:30 today. And I want to thank the NIH for sponsoring or supporting some of the studies that I've shown you this morning. Thank you very much.